Hello dear, Assalamu alaikum and welcome back. This is Dr. Hadi here. Today's topic is from the Biochemistry, a very interesting topic, that is isoenzyme. In today's lecture, we will discuss some important headings like the concept of isoenzyme. What is this isoenzyme? What is the idea behind isoenzyme? Why it is present in our body? The existence of the isoenzyme, how these are present in our body, why these are present in our body. And we'll also give you an example of isoenzyme by describing the concept of lactate dehydrogenase LDH. And we will also discuss the significance of the different catalytic activity of these enzymes in the example. And we'll also discuss with you the diagnostic importance of the LDH or the isoenzyme. So actually the board, this time the whiteboard looks filled. Actually I have recorded a video of around about 35 to 40 minutes, but when I realized that the mic was off and even its battery was also lost, battery was discharged, so I replaced another battery and now the microphone looks okay, the power button is on. So that is why I have to definitely deliver a lecture once again. And one thing more, for those students who can't really understand English, means they feel difficulty in, don't worry, this topic will also be uploaded in Urdu as well, but this time in English, and next it will be uploaded in Urdu. The Urdu video lecture will be uploaded in Dr. Hadi Academy's channel, and the English version of this video will be uploaded in Medical Globe by Dr. Hadi. So let's start the concept of isoenzyme. In order to understand the concept of isoenzyme, I would like to tell you that let's suppose we have a reaction where the A is converted into B. That's just a reaction. And this reaction is catalyzed by the enzyme. I have mentioned it as E, a circle E. E is what is the enzyme. If you see, there is black color and the circle is there. The same reaction is again catalyzing by another enzyme. This enzyme is catalyzing the same reaction, but this one is different. Look, here it is in the red color, here is the black color. It is spherical, it is square. So just to tell you that these are two different enzymes, but they are catalyzing the same reaction, A to B. This kind of enzyme which are catalyzing the same reaction, these are called isoenzyme or isozyme, simply isozyme. And more, actually these two are the multiple form of one single enzyme. These are just actually one single enzyme is there, and this and this one, these two are the multiple form of the same enzyme. So we can E and E. Okay, fine, this is got. If just like here in this example, these two are the two multiple form of the enzyme isozyme. These are isozyme of one another. There are some enzymes who have 5 multiple forms, 6 multiple forms, 3 multiple forms, whatsoever. These multiple forms are called isoenzyme. Now, can you see the definition? Multiple forms of an enzyme catalyzing the same reaction. Look, this is very important, the same reaction. These, okay, now these multiple forms, either these are different or similar. So one thing is for sure, from the diagram you can see that these two enzymes are different. Okay, these two are definitely different. In what other aspects these are different? The two enzymes may have different pH. The two enzymes may have different temperature. The two enzymes may have different affinity to bind with substrate. Different affinity. And the two enzymes may have different movement. One enzyme will move slowly, especially in the gel, electrophoretic gel, and the other form may move fast, with a faster rate. So what I would like to say, what I wanted to say actually, that these multiple forms are different both chemically and physically. Even one may be acidic, other may be basic. So this is for sure, we must know that. Now, where are these present? How are these isozymes are produced in our body? It's very simple. There are two ways by which we can understand this. Number one is the chromosome level. This is our chromosome. This is what? This is our chromosome. The one in the red. This is a chromosome. And at the chromosome, these small segments are gene. Gene number 1, gene 2, gene 3, gene 4, gene 5, 6, 7, 8, 9, 10. There's different genes are there. Okay. So, one gene, let's suppose this is the gene. This gene is producing enzyme. This one. And here, another gene we have, this gene is producing another enzyme. This one. So on the chromosome we have different genes, two genes, they are producing two enzymes, and these two are the isoenzyme. Maybe if there are 3 genes, then they give us 3 enzyme, 4 enzyme, and all of these will be the isoenzyme. So this is the number 1. Number 2, Golgi body is processing. Golgi body is processing. What is this? You know, inside our cell we have different organelles like mitochondria, like nucleus, like lysosome, like endoplasmic reticulum. But one is the Golgi bodies, Golgi complex. This is the diagram. And you know, the purpose of Golgi bodies is what? To process the incoming protein. Let's suppose this is what? Protein. It is synthesized on the endoplasmic reticulum. Endoplasmic reticulum. Okay, so on the endoplasmic reticulum, the protein is processed and now it will come towards the Golgi complex. Protein is actually of the enzyme. Okay, these protein will give us enzyme. And now this protein will be in small vesicles, small sac-like structure called vesicles. So these vesicles enter into the Golgi complex, Golgi bodies. Here some of the carbohydrate, look at the black dots that I have mentioned here, black dots. The black dot is actually CHO, carbohydrate, or simply carbs. So carbohydrates are mixed with these protein. So protein and carbohydrate are mixed together. Now this processed material will come out, like we can say this is the processed material number 1, and this is our enzyme 1. Okay, now next time another protein will come here, or you can say the same protein comes here, and once again carbohydrate is added. And this is another processed enzyme. These two are same enzyme. Here carbohydrate is linked, here also carbohydrate is linked, both. So but the carbohydrate here is more, but here the carbohydrate is less, making these two different. Protein is same, but the carbohydrate content is different. It has high carbohydrate, it is less carbohydrate. So these two enzymes are different enzymes. These are isoenzymes. So this is another way. Now, I would like to give you one example of isoenzyme, that is lactate dehydrogenase. Lactate dehydrogenase is a very, very famous enzyme. And this enzyme has 5 different multiple forms. Before we discuss the multiple forms, I would like to tell you what is the role of LDH, lactate dehydrogenase. Lactate dehydrogenase, as the name shows, this enzyme is involved in the conversion of lactic acid into pyruvic acid and pyruvic acid back into lactic acid. This is just the role. And you know, pyruvic acid is the product of glucose in the metabolism of glucose, provide us energy and so on, Krebs cycle and this and that. We will not discuss this in today's lecture. Okay, so this was just the simple role. Now if you come to the structure of LDH, lactate dehydrogenase enzyme. Lactate dehydrogenase enzyme consists of 4 units. 4 units. 4 units are there. These 4 units are called as subunits. One LDH enzyme has 4 smaller units. So we can also call as a tetramer. Tetra means 4. Tetramer. There are 4 units are there. Okay. And these 4 units are mentioned in the form of ABC little h or m. If you see here, there are 5 multiple forms of the lactate dehydrogenase now. This was just the structure of just one lactate dehydrogenase. Now, LDH1 is the first isozyme, LDH2 is the second isozyme, LDH3, LDH4, LDH5. Okay. Now, first come to the LDH1. LDH1 can be written as H4 only. What does it mean by H4? H4 means there are 4 units. And H means that this enzyme is predominantly present in the heart, not in other body parts, predominantly in the heart. However, little bit is also present in the red blood cell as well, but predominantly it is present in the heart. H-E-A-R-T, H. In the beginning of heart there is H, so we mention it H4. Now H is for heart, then what does it mean by 4? 4 means there are small units, small, 1, 2, 3, 4. 4 smaller units, these combine to give us H4 or LDH1. And these all subunits are similar, H, 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 H. Now if you come to the LDH2, in LDH2 we write it H3M. How? We write it H3M. H3M means there are 3 H type of subunits and 1 M type. Because look at that, I have mentioned it is in the red color and even the sphere. 1, 2, 3, 4. Again 4 units are there, but 3 are H type and 1 is M type. So H can be written as as a blue square boxes, heart, blue square boxes, and muscle as a circle, red circles, in the form of red circles. So here you can see these are the this is the LDH1 and this is the LDH2, which we can write H3M. And now where is LDH3? LDH3 can be mentioned as H2M2. What does it mean by H2M2? In H2M2 means there are 2 H units, 2 blue circles, squares, and 2 red circles. This is H2M2. Now you can write the fourth one. The fourth isoenzyme is LDH4. LDH4 can be mentioned as H1M3. It is the diagram. LDH5 is the 5th isozyme. You can write simply as LDH5M4. There is no H. Just like here, there is no M unit. Here, there is no H unit. It is purely M. And here, M. This is purely present in the skeletal muscle. So this is predominantly present in the skeletal muscle, and this one is predominantly present in the heart. So 2 different, heart and muscle. Now one of the enzyme in the heart has its different function, and the other enzyme in the skeletal muscle has different function. We'll discuss this significance of different catalytic activity. Why? What is the significance of this? We will not discuss the function of other isozymes. We will just discuss the function of LDH1 and LDH5. Only these 2. You will get something. In heart, if you come in heart, inside heart, the LDH1 is there, present, but it cannot convert pyruvate into lactate. So pyruvate cannot be converted into lactate, and because of this lack of the lactate, fatigue is not there in the heart, in your heart. Just like when you are working on the skeletal muscles, you are performing exercise, heavy exercise, you are pushing loads, heavy loads, and at the end, your muscle will get fatigue. You will feel some kind of thrashing pain. That's called lactic acid pain, lactate. But your heart is working 24 hours throughout your life. There is no fatigue because there is no lactic acid formation in your heart. That is why, and why this happens, because pyruvate is not converting into lactate. Okay, this is one. Now, LDH1 actually inhibited by the pyruvate. Pyruvate will inhibit the enzyme. Pyruvate will not allow the LDH1 to form lactate. It is in the heart. But if you come into the skeletal muscle, it is quite opposite. In skeletal muscle, the pyruvate can be converted into lactate. Here, pyruvate is a friendly substrate. Pyruvate can bind with LDH5. Pyruvate can bind. Here, pyruvate cannot bind. I'm supposed to say, I hate you. Pyruvate cannot bind LDH1. So that is why it cannot convert into lactate. But here is very friendly. Pyruvate is very a friend of LDH5. Here, the pyruvate is converted into lactate. So in muscle, lactic acid can be formed if the condition is anaerobic. If the condition is anaerobic, means if oxygen is less, you can feel the fatigue. Here is fatigue, here no fatigue. So we've got one most important thing, that why your muscle can be felt in the pain form. Muscle can be fatigued, but your heart muscle cannot be fatigued. Instead of its continuous strenuous exercise. Now the diagnostic importance of the LDH. Is there any diagnostic importance of the LDH? Yes, we say yes. Why not? We can predict some disease by just taking the blood sample from the patient and checking the amount of enzyme. We can predict either the disease, the pathological condition is there or not. For example, before I go to the topic, I would like to tell you that inside your blood, you have the 5 different isozymes in your blood, in my blood, in your blood. H1, LDH1 is 25%. LDH2 is 35%, LDH3 is 27%, LDH4 is 8%, LDH5 is 5%. Okay, by graph, if we see that this graph, an intermediate peak is there for LDH1. This is for LDH2. Why this peak is little bit higher? Because 35% is there. So this is 35%. This is 27%. This is 8%. And this is very small peak. I should like the small peak is there. 5% is there. 5%, 4%, 3%. This is a normal. Now, there are two peaks. If you look at this graph, what will you see? What will you observe here? So you will observe that the lactate dehydrogenase 1 is more than lactate dehydrogenase 2. Normally 2 is more than 1, but in pathological condition like heart attack, myocardial infarction, MI, myocardial infarction, the LDH1 is increased than LDH2. So this shows that the patient has myocardial infarction. Okay, this is one. If you come to this graph, here this graph is quite similar to this one. 1 is less, 2 is high, 2 is high, 3. But here the 5th peak is high than here. So it means that LDH5 is increased. This patient 1, patient 2. Patient 1 has myocardial infarction, MI. Why? Because H1 is high. Patient number 2 does not have myocardial infarction because his or her H1 is okay, but the LDH5 is very high. When the LDH5 is very high, this means that this patient has liver disease. Which disease? Liver disease. So diagnostic models of LDH, lactate dehydrogenase enzyme, we can predict lots of disease as well. So in today's interesting topic, we have discussed the main idea of isoenzyme, that these are the multiple forms of the same enzyme. And then how these are existed, they are either formed from the different genes on the chromosome or from different processing aiding of carbohydrate in the Golgi complex. And example of isoenzyme is lactate dehydrogenase. Lactate dehydrogenase has 5 different forms. Each lactate dehydrogenase has 4 smaller units. These 4 smaller units may be all H type or M type or H and M mixed type. And then we discussed the significance of differential catalytic activity, that in the heart pyruvate cannot be converted to lactate because pyruvate is the enemy of the LDH1. Pyruvate do not allow the LDH1 to convert, to form lactate. But in the skeletal muscle, pyruvate can be combined with the enzyme. Enzyme can convert pyruvate into lactate. So this is the significance. And then diagnostic importance, inside our blood, serum, different concentration of the isoenzymes are there. By checking, analyzing the quantity of the enzyme, we can predict either a disease is present or not. I hope you got this topic. As I always say, if you got the idea, share the lecture, share the video with among your friends, and give me a smile on your face. Bye-bye.